Great. Okay, so thank you, Sven, and thanks to the organisers for giving me this opportunity to speak today. And this is going to be somewhat of a contrast to the previous talk you heard, because we're going to effectively zoom right in at the lowest level that you can really think of that's important for neurobiology, and that's right at the molecular level. And I think the next three talks are going to focus on that. At the very basis of most molecular signaling are membrane proteins, and you can see in the background here's an example of that, just sort of embedded in a typical membrane simulation environment, if you like. So the first thing to really put things in perspective is to sort of consider how you might classify membrane proteins, and you can do that in a variety of ways. You can simply look at the sequence and look at the phylogeny, and there are a variety of databases that try to do that. An example of that is this transporter classification database. That, however, is a little bit prone to some errors because we simply don't know enough about the structure at this stage to make very good statements about whether they're in the same class or not. The other way to do it is to do a rather sort of opposite almost, and try and just consider the sort of basic or overall fold that you think these proteins might have. And they basically fall into two sorts of categories. They can either be a barrel or they can be an alpha helical. And actually by those two overall folds, they have, there's an inference about their location. You only find the barrel proteins in the outer membrane of bacteria and of mitochondria, whereas plasma membranes will have helical proteins. But we can do a little better than that now. Recent structural progress now means that we can do something a little bit more sort of fine-grained, if you like, and we can now try to sort of classify proteins and membrane proteins by their sort of known structure and their function. And this is just an example subset of that we can do now. Now, most of this has been possible due to really quite good advances now in structural biology, and this is an example from Stephen White's recent review last year. No structural biology talk is complete without one of these sorts of curves, which shows you the sort of growth in structural information that's coming along. And this actually mirrors what happened for soluble proteins sort of 25 to 30 years ago. And now probably there's about over 200 unique structures solved. And probably about 25 years ago there was only a handful. So this is really very good. This is the good positive news that's coming out in recent times. So here's some examples sort of proteins that that are coming out these days. One of the first ones, well, not one of the first, but the first ion channel that was solved was a bacterial potassium channel, KCSA, that was solved in McKinnon's lab in '98. And then we have a mechanosensitive channel, the first sort of TM structure of the nicotinic acetylcholine receptor, which is very important, of course. And that was followed by a refined version of that. And we have then things like the NAC channel and the acid-sensing ion channel. And you can see here, they're all helical in the plasma membrane. Then we have bacterial ligand-gated ion channels. An example of that is this L-GIC channel, so-called. It looks very much like the nicotinic acetylcholine receptor, but there are some important differences. One important class is this, the GPCR protein class. The GPCRs, of course, are very important from a drug discovery point of view because they're well over 50% of all known drug targets. So structural information about GPCRs is going to be increasingly important. And I think you're going to hear a little bit more about the sort of underpinning dynamics in the next talk about GPCRs. One of the most recent structures is the glutamate receptor structure, which came out last year from Eric Guo's lab. And this really is a big structure. You can see here, this is an enormous structure. Quite simple architecture in the membrane, but quite a lot of modulatory sites outside of it. And I'm going to use this actually as an example protein to sort of describe a couple of key points, or about a couple of key points, the sort of general things that are emerging from the structures that are coming out. So the structures are great, but they're really only a starting point in some ways. A lot of these proteins that we're talking about that are embedded in the membrane are very dynamic. They move by definition. They have to move in order to conduct the signal. You know? So you could say, well, why don't we just solve structures for all the different states using crystallography? Well, although there was great progress that I showed you in those previous slides, it's still very difficult and time-consuming to do structural work in the membrane. And so this is really where computational methods can provide key insight. We can provide some sort of nice complementary stepping stone in between to provide information about how these proteins move. And we can also begin to ask sort of questions about the underlying basis for which they work. And through that we can therefore direct further experiments that can be done, and indeed can direct crystallography, for example, in a more targeted way perhaps, to try and get the information that we need. And an example sort of illustrates this key point, these 2 points really, is the sort of potassium channel, which was first solved over 12 years ago. But the physical basis for selectivity is actually still rigorously debated today in lots of the literature, biophysical literature, okay? So although the structure was solved, you might have thought, well, it's fairly obvious how it works, but actually turns out it's a little bit more complicated than we first imagined. So going back to that structural information, there are a number of sort of themes that you can pick out that are starting to become clear about membrane proteins, and particularly signaling proteins. The first of these is helical dipoles. So although there's lots of helices in the actual overall structure, they also play a key role in actually sort of stabilizing things like ions in key positions within the membrane, for example. Another example is regulation by small ions. So although a lot of these proteins do conduct ions, they are also regulated by ions as well. And I'm going to talk about that in a little bit more detail in a second. Another emerging thing is things like hinges and switches. A lot of them tend to have sort of key motifs. For example, potassium channels have proline, valine, proline as a motif that is used to act as a sort of gate, or a hinge in the gate rather. And things like ionic locks and so on and so forth are important sort of aspects of switches, which are generally starting to come out as well. The role of symmetry is quite interesting. For transporters, you typically have two-fold inverted symmetry type structures, and in the sort of channel structures you tend to have tetramers or pentamers or higher order oligomers than that. And that can also be an important role in terms of their function. And finally, one thing which has been really neglected in recent years is the role of the lipid. It's sort of just tended to be thought of as some sort of non-polar sort of milieu, but actually you need actually very precise lipids in order to activate some of these proteins, okay? And if they don't have the right lipid present, then actually the protein won't work. So we're going to have to start paying attention to this component in more detail in the near future, I think. So I'm going to just sort of illustrate some of these points just by reference to the glutamate receptor. In particular, I'll talk about regulation by small ions and a switch that we found also in this structure. So just to put it in context, I'll just sort of come back to the structure, which is here on the right. It's a tetrameric protein, okay? And it's made of, each monomer has a rather complicated sort of topology, if you like. So you can see here that the chain that forms this domain here, this is the ligand binding domain, this is the business end of the molecule, okay? You can see here that this is formed by a discontinuous chain, so it's actually a rather complicated structure. And you might sort of recognize this if you're in the field. This is sort of similar to an upside down potassium channel, okay? So that's kind of useful for us to keep in the back of our mind. And now one thing that's quite interesting about this molecule as well is there's a symmetry mismatch. So in the membrane we have true fourfold symmetry, whereas upstairs, if you like, it's really arranged as a dimer of dimers, okay? So that poses interesting questions about the activation mechanism of that. How do four of those glutamates bind in order to activate the channel? And we know, as I said, that these proteins move a lot, and we know sort of roughly how they move. Okay, you start off in some resting state. Here I'm just showing you one pair of dimers, if you like. So this is the ligand binding cleft only, and I'm just showing you the M2 helices, which are the bits that line the pore, essentially, okay? So you'll start off in a resting state, and you bind some glutamate like this, and that binding event allows you to do mechanical work, which allows this cleft to close, and it pulls open this M2 channel, presumably. We don't know that for sure, Okay? And then after a while, of course, these channels undergo desensitization, which you all know. And that is thought to reflect a sort of reorganization of this dimer interface in a way that we don't really understand at the minute. Yeah? So I'm going to sort of focus in on that, but I'm going to use molecular dynamics, and I won't bore you with the details, but essentially we just solve one equation rather repetitively. There's this equation here, Newton's equation of motion. And in order to do this, we describe all the forces on the protein using a sort of force field. And we start with some initial atomic position, some initial description of position, and that's usually an X-ray structure in our case. And after you integrate this many millions of times, of course, you end up with a trajectory which describes the positions and energetics of all the atoms over time. Okay? That's the basic principle. I mean, there are obviously lots of modifications on top of that. So, as we've been hearing, the sort of scale problem is also a problem for us, even just to try and look at the sort of dynamics of these things at an atomic level. So, you can see here, if we're just sort of looking at the channel section, you have a reasonable size box. And if you want to look at channel activation, you probably have to include the ligand binding core. But if you want to look at the whole receptor here, we're sort of getting on for 1 million particles if you have water, explicit water around the system as well. So that's often a problem. And there are numerous ways around that. One is actually to sort of be a bit smarter about what you simulate, and we can do that in this case because it's a very modular structure, so we can sort of work with the domains and look at those individual motions, if that's appropriate, and use that as a method. So we chop up the system. And indeed, I'm going to sort of just focus really on this ligand binding core initially. You can also use other methods to get around this problem. There are a lot of very technical methods that you can use. Non-equilibrium simulations, coarse graining approaches where you represent more than one particle. You represent something like 4 particles by 1 particle instead. Or you can do something like biased sampling and correct for that bias afterwards. Okay. So this, I'll now describe an example of regulation by ions. So in one category of glutamate receptor, the kainate receptors, it was known a few years ago that cations and ions regulate the desensitization rate of these receptors. Okay, this is work by Mark Mayer. And this anion site was known to be located right at the top of that dimer interface. Remember, this is the interface that rearranges upon desensitization. Okay? But at that point, the cation site was still elusive. There was no information about where it was, despite lots of hard work, actually. So the first thing we did was just simply do a simple electrostatics calculation. This is the ligand binding core you're looking at side on. And if you just sort of map on the electrostatics calculation on top of that, you can see here on top there are 2 rather intriguingly nice two red regions which indicates nice negative pockets. Okay, so that's the first clue that they might be there. And the second clue was to use a very simple program called GRID, Peter Goodford's GRID program. Essentially you put the protein onto a grid, and then you put your probe molecules or atoms at different points on that grid and calculate the interaction with the protein. And you can see here nicely that the cyan is the prediction here, and you can see it nicely reproduces the chloride site, and then it's got 2 sites for what it predicts as sodium density. And in the crystal structure, these are actually occupied by water molecules. Okay, so at this point we thought, well, let's go ahead and stick a sodium ion in those sites and see what happens. Are they perfectly capable of holding a sodium or not? And could we, on the basis of that, then make a prediction to get our crystallographic colleagues to perhaps try a bit harder. And sure enough, if you do a molecular dynamics simulation of sodium in the site, so sodium are the yellow balls here and the chloride is green, it just wobbles around nicely, nothing too much happens. It's quite a boring simulation. Conversely, if you put cesium in the binding pocket, then it wobbles around, but after a while the cesium is not very happy and it will kind of drift out. Okay, so you'll see this probably happen with this top one in just a second. This ion here, oh, there we go, not very happy, so it just kind of floats away. And that's quite reproducible, still not very quantifiable at this stage. So at this point we decided to do free energy calculations. We can calculate the free energy of binding an ion to those different sites. We can do that absolute free energy for the binding of a sodium ion, for example. And then we can also calculate, using this sort of method, a thermodynamic cycle, relative free energies. Relative free energies are a bit easier for us to calculate. The idea is that you calculate the energy alchemically. This is something you can't do in the lab, but you can do this computationally. You calculate the free energy that it takes to convert an ion from, let's say, a sodium into a potassium ion, and you do the same calculation basically in the protein. So this is in the solvent and this is in the protein. You can then solve this thermodynamic cycle, and you therefore get out relative free energies of the different ions. And if you do that, then you end up with a relatively straightforward cation affinity series. Okay? So at this point we were getting a bit nervous because we were quite convinced that there should be sodium ions in that pocket, and there were several crystal structures that were out that didn't have sodium ions in there. Okay? So at this point we went back to the crystal data. If you go back to the crystal data, this is the 2F34 structure, and if you expand the crystal lattice, so the green structure is the normal dimer, if you like. If you expand the crystal lattice, what you see is that there's a loop of a neighboring molecule in the lattice that's basically dipping a lysine, and it's dipping its lysine into our predicted pocket. Okay, so that nicely accounts for why in that structure they didn't see sodium or any other ions, basically. Okay. Similarly, in this structure, this is another structure that was reported, done in very high concentration of ammonium sulfate, and in this this structure, they reported a water molecule located here, but actually that's more likely an ammonium ion. Okay, it's very difficult to distinguish between ammonium ions and water molecules in crystallography. Okay, and similarly, if you go back and look at mutation data, if you make this methionine into a lysine in kainate receptors, then you actually abolish the sensitivity to cations. You can change the cations, you can change the concentration, nothing much happens, okay? And that can easily be reconciled just on the basis of steric interference, if you like. Imagine if that's a lysine, that's obviously not going to be too happy sat next to another lysine, okay? Sat next to a sodium ion. So, just as we were doing that, then our collaborator at the NIH, Mark, managed to get the crystal structure. This is actually with lithium in it, and to our relief, there was lithium ions exactly where we predicted. So that was a rather nice confirmation of the molecular dynamics experiments, if you like. And we can go a little bit further and say, well, how does that correlate with the experimental information? Because experimentally, they weren't really measuring affinity of those ions. They were actually just measuring the desensitization rate. So the desensitization rate is shown on the left here. You can see that the sodium ions desensitize much slower than, for example, the cesium. Okay, and if you plot basically that desensitization rate with respect to sodium on the x-axis against our affinity, then you get a nice linear correlation. Although that makes intuitive sense, there's actually no reason why you should expect that. Okay, we're just measuring affinity and they're measuring desensitization, a sort of effective process. Okay, but it's nicely reconcilable on what you know about the interface, where those ions sit. It obviously makes sense that they kind of act like a kind of glue. So you can do other things as well in the computer that you can't do in the lab, and you can examine things at a very basic level. You can start to ask questions about the fundamental aspects of the selectivity. So for example, here we can sort of play and work out which bits of the binding site are responsible for it being selective for sodium. Why is it suddenly selective for sodium and not potassium, for example? And we can do that by basically playing around with the charge on the carboxylate group of one of the aspartic acid that's coordinating the ion in the binding site. And if you just do the calculations, again, the free energy calculations to transmute it from a sodium to potassium, and you gradually just sort of change the charge contribution on this carboxylic acid down to something that looks more like a carbonyl charge distribution, then you can see when you get to that point, actually this site becomes non-selective. Okay? So in the last couple of minutes, I'll just talk about something else. The ligand binding domain, of course, is the workhorse, you know, and we can move between these two states, and we can do that. That's just a morph between the closed and open states of the open cleft and closed cleft of the crystal structures. But actually, how does it work? You know, what's going on? How does it move? Is it torsions that change? Is it something else that happens? And we can observe this actually directly through simulation. What's going on? This is one of those clefts looking side on, and in these clefts there's basically a hydrogen bond between, in the open cleft form, between the glutamic acid and this lysine here. Okay, and then when that lysine breaks that hydrogen bond and reforms it with this aspartic acid, that is what allows the cleft to close. Okay, and this is totally reproducible, and you can indeed do a simulation where you constrain that hydrogen bond to be fixed, and you can prevent the cleft from closing. Okay, so this is a sort of switch, a molecular switch, an ionic switch, and that's something that's been seen in GPCRs, a so-called ionic lock, which has also sort of been studied quite a lot by simulation, which was unexpectedly broken actually in the crystal structures. Okay, so I'll just sort of end with some challenges that I think are pertinent for the field. And one thing is the sorts of things that everybody's been talking about at this conference, of course, and that's kind of joining the gaps between the descriptions. So we're very much at the bottom. So how do we feed our information that we've got here into the higher level models, into coarse grain models, into differential equation models, etc. So-called multi-scaling modeling. And the good news is that there are lots of people doing that. This is a very active field in biophysics at the current time. What's less perhaps in vogue, but is still a major problem, and that's accurate prediction of binding free energies of small compounds. So even if you know how your system works, right, in very great detail, you still want to really be able to predict how a certain compound will bind to that and how it will interfere with that, yeah? So that you can, you know, and as we heard this morning, you want to disrupt those networks, right? So, but you still want to be able to do that in predictive nature. And this is fundamental to that process. And so related to that, and sometimes more difficult, but sometimes easier, is the prediction of conformational change. And obviously for signaling, that's obviously very, very important. So I'll just end with the acknowledgements. Just Ranjit is responsible for most of this work, and my colleagues at the NIH, and these people for funding, and you for your attention. Thanks.
Well, thank you for this very interesting presentation. I think that the question about joining the gap between modeling at different levels is something that we could return to in the panel discussion. It seems to be an obvious point that needed a wider discussion. It's one thing that is quite, I don't know if it's clear to everybody who's not directly involved in the field, but one of the major ways that this has been working is that all these proteins, all the structures and the models that Dr. Biggin showed us had been cloned before the crystal structures were sold. Isn't that correct? So you always had the amino acid sequence. And with modeling, molecular modeling, you had at least theoretically a possibility of making a model. So it must have been quite rewarding for you in the case. And I had to admit that we have also had a few cases of the same in our group. You make a model, and then the crystal structures come out. You hear that it's published, and you are sort of a little bit in trepidation before you see it. And then if you feel that you see it confirm your models, oh, that was good, because you can never be sure. Thanks. Questions, comments to this paper? The obvious thing about these kind of studies, the key word, the questions that we have been asking is concerning molecular mechanisms. Do you agree? That is the interest.
I think the fundamental interest for us is very much what are the fundamental mechanisms operating at that level.
Yes.
And that's something we still really do not understand in that much detail at the atomic level.
Yeah. Well, I was supposed to be a chairman, not a discussant all the time. I have several other comments, but I invite you. Yep.
Well, just a question with respect to your last point. What is the fundamental problem in calculating the deformations of a molecule, in a sense? You can, say, displace any molecule, any atom in a molecule and look for the consequences for the binding energies and the free energies. Is it a combinatorial problem in the sense that you can perturb one or you can perturb two or any subcluster of atoms in the molecule? Is that a problem?
The problem for us is really essentially a sampling problem as much as anything. We're still at the stage where we cannot run the simulations long enough to make accurate predictions about which conformations are going to predominate, that you would pick up in the experiments. It doesn't mean the conformations aren't there, it just means that the ones that you see in the experiment, of course, are usually a time and space averaged over much longer time and space than we can simulate at the minute. Yeah. So that's the fundamental problem. There are ways around that technically, but they involve huge computation as well. You can get the right answer, well, roughly, but that involves a lot of computational work in itself. So there's no clear path to doing that regularly, shall we say.
Okay. I'm not so familiar with this point, but to what extent do quantum mechanics is involved in this type of calculations?
Okay, so there's no QM here at all. We don't use QM. It's all just Newtonian physics, essentially. You use QM typically, though, to parameterize the charge. So okay. the charge that you represent one ball, if you like, one atom with, that is calculated typically by using a QM calculation. So you have at least a rough idea of that it's right, that it reproduces the electrostatics of that small fragment, shall we say.
Okay, thank you so much.
So the force calculations are what we call molecular mechanics, which is sort of simplified way. I think I would like to add one comment. We heard from Dr. Biggin that I think it was a metabotropic glutamate receptor that has 800,000 atoms. So for each atom, when you want to do a simulation, you have to have the 3 coordinates in the space, x, y, and z, and then the velocity. But without being too technical, I think it's fair to mention that the time step you calculate in order to make these calculations meaningful, at least we use 0.1 picoseconds. Mm-hmm. And that means that you have to do 10 to the 13th calculations in order to have a simulation of 1 second. 10 to the minus 13, that's a lot of zeros. And that's why we have these limitations that Dr. Biggin mentioned at the end. It's difficult for the sheer number of data that you have to generate and treat and present to do calculations over more than nanoseconds. Maybe now I saw some of the next speakers. One of his posters he did in microseconds, 1 or 2 microseconds, but that really seems to be the limit. How long are your simulations going on, the longest that you've been doing?
So typical simulation times for these sorts of systems are about 30 nanoseconds.
Yeah, that's what we do also. Yeah. Any other questions, comments? Okay. Thanks again.